Do you know who is presenting the case? Any of you?
There's no case.
No case. So I've got no safety net at all, is that what you're saying? Do you guys know if Dr. Joplin made it in last night?
Yes, he was here 2 days ago.
2 days ago. So he's been here.
We suspect his rental funds are I'm getting hand signals from the back that suggest that the second time in 24 years of having a speaker not show would not be occurring today, which I'm pleased with. Oh, at least Dr. Belknap is going to do the thing if nobody else is. A couple of things. One, you have 12 more days to submit your vote in this year's election. It's another important part of our fiscal and social responsibility. Number two, I hope that all of you have had some conversation with your front office staff and your medical assistants about what questions you're asking when someone presents with their child with fever, nausea, vomiting, diarrhea, because it is part of the growing effort to make sure that we identify folks who have come into our facilities from North Africa. At this point, I think that Ben is ready to begin the introduction for our ethics conference.
Thank you, Tom. I was just trying to see if I could take a couple of years off your life by showing up.
He was going to do it.
The main thing that I have to do now is actually just get our our slides queued up.
So I should do filler while you
No, this will take me 2 seconds. I just have to hit the. What I will do is I don't want to do too much multitasking because I don't want to. I'll wait till I get things ready. So this is our 8th annual Truman Katz Lectureship. And before I introduce our speaker, Steve Joffe, I want to say a few words about Truman Katz. And let's see what I have to do here. So Truman was the CEO of Seattle Children's Hospital for 26 years. When he retired in, let's see, what's going on here now? What's that? I just want to get onto my O drive.
It's okay. Stand by. Go ahead and speak.
Okay. So when Truman Katz retired in 2005, the Board of Trustees both endowed the center as well as this lectureship to commemorate him. And often Truman is actually here in attendance at Grand Rounds every year, although we did this a little later, so he's already moved on to his winter home in Palm Desert, although we did see him a few weeks ago. And Truman has been a very strong supporter of ethics and the center. And you know, what's also been very interesting for me is I travel around the country and I tell them I'm from the Truman Katz Center. He himself is known around the country, and so it's often very interesting the way I hear stories about him. During his tenure as a CEO, was responsible for a lot of major innovations at Seattle Children's, including working to develop the Seattle Cancer Care Alliance, the collaboration between Children's UW and Fred Hutch, among a number of other very important collaborations. Okay, oh, great, fantastic, okay. So, actually, a different one. Thank you so much. That was.
So this is the reason I encourage us to arrive on time.
Oh, yes.
I know if this were CNN, you would be happy to do some other discussion at this time.
This was, you know, what can I'm very sorry, Tom. Anyway, let me introduce, so I take full responsibility for this breach of protocol. But it's really my pleasure to introduce, and this will be a very memorable Grand Rounds for that reason and nothing else, but more importantly, it'll be memorable because of Steve Joffe. Steve is a colleague who I've worked with for many years. We have a lot of overlapping interest in both issues related to genetics and research ethics. Steve is an oncologist. He, up until a few years ago, was at Dana-Farber and Boston Children's, and he recently moved to University of Pennsylvania. He's the vice chair of the Department of Bioethics and Health Policy, and as the vice chair, he is responsible for their bioethics division, which is a very vibrant bioethics program in a major medical school. They recently have started a fellowship, and Steve is also the director of that fellowship program, so we now have some competition with our fellowship program. I think with that, I'll stop since we're running late and welcome you, Steve, and to your talk.
Great. Thank you, Ben. It's a pleasure to be here, and I hope you all had as exciting a start to your morning as I did. So hopefully this will live up to the excitement of so far. I'm going to be talking about the ethics of early phase trials in children with cancer. And I hope some of the lessons, I don't know how many of you are here from a cancer perspective or work in oncology, but I hope that some of the lessons will be relevant not just in cancer, but in other areas of pediatrics and medicine as well. I have no financial relationships to disclose. I'm going to use as an example a clinical trial of a drug called Chrysotinib, just as an example of some of the issues that we're talking about. It's FDA approved for other things, but not for the treatment of kids with cancer. What I'm hoping to do is to analyze with you the risks and benefits of phase 1 trial participation in pediatric oncology, and to develop a strategy or think about a strategy for seeking consent to early phase cancer trials and really to early phase trials in general. So I think most of you probably know that phase 1 trials address primarily dose and toxicity. If you read the aims or objectives of a phase 1 trial, they're about dose and toxicity. Malcolm Smith at the NCI and colleagues a number of years ago said the primary objective of pediatric phase 1 trials is to define a safe and appropriate dose and schedule for new agents that can subsequently be used in phase 2 trials to test for activity against specific childhood malignancies. For most agents, the highest tolerable dose should be used to evaluate anti-tumor activity. They might, if they were to write this again, they might actually say this a little differently in the sense that we're now moving into molecularly targeted agents where the highest tolerated dose is not always the dose that you want to take forward, but nonetheless, this maximum tolerated dose paradigm is an important one that you'll see in the discussion. These are controversial kind of trial in general and particularly in pediatrics. You can go back to 1982, where Mortimer Lipset wrote on the nature and ethics of phase 1 clinical trials. And this is the first time that I've been able to find somebody who's really flagged the ethics of these trials as controversial. Ethical and practical concerns abound. Arguments have been made that these are, quote, not just for safety anymore, and that therapeutic intent in these trials is a justifiable objective, and we'll have a discussion about that. And as Rick Kodish wrote about 10 years ago, pediatric ethics and early phase childhood cancer research, the goals are conflicted, and we have lots of questions about the prospect of benefit, and we'll get into that. So here's just as an example of a trial, one that I think raises a number of interesting issues, for those of you who don't have firsthand familiarity with these trials. So this was published in Lancet Oncology last year, study safety and activity of Chrysotinib for pediatric patients with refractory solid tumors or anaplastic large cell lymphoma, a Children's Oncology Group phase 1 consortium study. So Chrysotinib, for those of you who aren't familiar with it, is an oral inhibitor of 2 kinases, ALK and MET. It's highly effective against ALK translocated lung adenocarcinoma, which I think is about 3% of all lung adenocarcinomas, and has actually been FDA approved and is really a remarkable drug for that subclass of adult cancer. Now, there are pediatric tumors that contain ALK variants, either translocations or mutations or amplifications, ALCL, anaplastic large cell lymphoma, a rare tumor called inflammatory myofibroblastic tumor, and neuroblastoma, which is one of our most common solid tumors. And so the COG was very interested in looking at Chrysotinib both in general and particularly for ALK mutated or ALK variant tumors. So they initiated a phase 1 trial. The goals were to establish the maximum tolerated dose and recommended phase 2 dose of Chrysotinib among children with relapsed or refractory solid tumors and ALCL, to define and describe the toxic effects of the drug, and to characterize its pharmacokinetics. And almost any phase 1 trial is going to have a list of primary objectives that looks like this. Also, as a secondary objective, it was mentioned of looking at tumor response and some other things. The eligibility criteria for this trial were broad. They involved patients between 1 and 21 years old who had measurable disease, either solid or CNS tumors or ALCL, refractory to therapy, and for whom there was no known curative treatment. And this is, again, a typical kind of eligibility for phase 1 trials. And because of the particular interest in these ALK or MET mutated tumors, there was an expansion group, a larger group, where kids who were known to have tumors with ALK or MET variants could enroll. And then a second expansion group for kids with neuroblastoma, whether or not they had ALK or MET variants. The study design, again, fairly typical phase 1 study design, up to 6 dose cohorts. The starting dose was 80% of the adult maximum tolerated dose, which was known. And this is common, or actually typical, in pediatric phase 1 trials, is most of the time we know the adult dose, and so we start the pediatric dose escalation just a little bit below the adult. And the maximum tolerated dose was defined at the highest dose level at which fewer than 2 participants in a cohort of up to 6 experienced a dose-limiting toxicity, which was defined in the protocol. So I'm curious, just by a show of hands, how many of you, just based on the very brief summary that I've given you so far of the background and the design, think that this trial offers children with recurrent or refractory ALK-positive tumors a prospect of direct benefit? Any hands up? Some hands down. Those hands that are down, you either aren't sure or maybe you think not. How many think it doesn't? Let's clarify what the hands down meant. I see one or two. So most of you who didn't raise your hands initially were uncertain. And in the front row, there was yes, benefit, but like this. What about children with ALK-negative tumors? Prospect of benefit, yes? They were eligible. Prospect of benefit, no? More no's and a bunch of uncertains, I think. Okay. All right, let's back off from this particular trial, talk about phase 1 trials in general. So many of you may know this paper by Zeke, Emanuel, and colleagues from about 14 years ago, where they argued that ethical research must satisfy 7 different criteria, that it must have social or scientific value, that the methods must be scientifically valid, it must be designed to get you to a valid answer, that subject selection must be fair, that there must be a favorable risk-benefit ratio, that it must undergo independent review by an IRB or some such body, that there must be informed consent, and that throughout the trial, before, during, and after, there must be respect for prospective and enrolled subjects. I'm going to argue that in phase 1 trials in particular, there are 3 issues that we need to pay attention to. The issue of fair subject selection, the issue of favorable risk-benefit ratio, and the issue of informed consent. And I will spend the rest of my time talking about each of these. So let's start with favorable risk-benefit ratio, which was sort of raised by this question of does Chrysotinib have the prospect of benefit for these participants? So I think there's really 2 distinct questions out here. The first one is, do phase 1 trials have therapeutic intent? And the second one, which is a different question, is do phase 1 trials offer participants a favorable balance of benefits over harms? And I'm going to spend some time trying to give you my view of these and show you some data. So the question of therapeutic intent in clinical trials is an interesting one. Up until about, I think, the 5th edition of the Declaration of Helsinki, there was a lot of emphasis placed on distinguishing research that did have a therapeutic intent from research that did not have therapeutic intent. And so the Declaration said, and it no longer has language like this, that there's a fundamental distinction between medical research, in which the aim is essentially diagnostic or therapeutic for a patient, and medical research, the essential object of which is purely scientific. Now, this distinction between therapeutic and non-therapeutic research has been used by some who wish to criticize, and actually some who wish to defend phase 1 trials. So Joel Frater and a colleague wrote in 2003, phase 1 trials are explicitly non-therapeutic in nature. Intended to reveal the pharmacologic action of the study agent, they seek to establish a baseline regarding toxicity. So the reason from the fact that their goals are about toxicity and dose finding and not about efficacy, to the conclusion that they therefore lack therapeutic intent. Pamela Sankar, one of my colleagues at Penn, wrote, while lack of benefit is the logical outcome of a phase 1 trial design, its absence remains controversial. Again, reasoning from the objectives of the study, which is not about efficacy, at least the primary objectives, to the conclusion that they lack therapeutic intent. My response to this, I actually disagree with this way of thinking about phase 1 trials, is that it's illogical to distinguish between therapeutic and non-therapeutic research. The primary objective or purpose of all research is to answer a scientific question. This is true of phase 1, it's true of phase 3, it's true of any clinical trial. That many clinical trials, many clinical protocols do have the prospect of benefit for participants, but it's always a side benefit. One we seek, one we try to maximize, but nonetheless it's a side benefit. Their purpose is to answer their scientific question, and then they may have, and we hope they will have, side benefits of a prospect of benefit for the participant. So their intent is to estimate dose and to assess toxicity, not to measure clinical benefit, at least their primary intent. But intent is not the same as consequences, and I think we can distinguish between these 2 things. So you can imagine a study that's intended to assess safety, but that may nevertheless offer a prospect of benefit. I recall a phase 3 trial that we participated in some years ago when I was in Boston, looking at the safety of a novel antifungal agent that was FDA approved for use in adults, the safety of this drug. The primary objective was what's the safety of the drug in children with fungal infections. But of course, there was clearly a prospect of benefit from these FDA-approved and frequently off-label used drug in children who had fungal infections. So objective was safety, but I don't think anybody would argue in that study that there was a prospect of benefit. And similarly, you can imagine studies that are designed to assess efficacy, but that may nevertheless lack a prospect of benefit. Think, for example, about a placebo-controlled trial where those assigned to the placebo arm are not going to get a prospect of benefit from that drug, even though the intent of the study is to assess efficacy. So then I think asking the right question, which is, do they offer a prospect of benefit? Here are the best data that I'm aware of. So there are a number of published meta-analyses. Here are the three newest ones. The first one of which is by Deborah Lee and colleagues from CHOP, looking at about 1,800 children participating in phase 1 trials. And what's reported here in this column is the complete plus partial response rate, basically percentage of children who had at least 50% shrinkage of their tumors. And you can see that for all studies, it was just under 10%, with about 3% complete response and about 7% partial response. This was lower in single-agent trials, which are the most classic phase 1 trials, and much higher in multi-agent trials, which often combine FDA-approved therapies with novel drugs. In this Horseman and colleagues, a much larger meta-analysis, which was limited to adults, also about 10% complete plus partial response rate. Single-agent trials were on the lower end of that. It depended upon which class of therapy they were looking at, or class of agent. And in multi-agent trials, again, largely higher response rates. And then in another meta-analysis by Tom Roberts and colleagues, which is actually not clear from this JAMA article whether kids are included in it or not, hence the question mark, limited to single-agent trials, complete plus partial response rate of about 4%. Toxic death rates attributed to the drug, incredibly consistent across these studies, always about 1 in 200, across every meta-analysis I've ever looked at. So these are the data. And you should be asking, so is complete plus partial response rate equal to clinical benefit? And that's the right question, and we'll talk about that. So that's some evidence that at least there is some effect on the tumor, which may, although we don't really know, translate into clinical benefit for participants. Here's the flip side of the story, which is that participants spend very little time on these trials. And the main reason they come off is that either their disease progresses or they have toxicity that takes them off. So you can see that ignore the multiple curves here, just look to average them in your mind. You can see that by 2 months after starting study treatment, only about 40% of participants remain on trial. Most have come off either because their disease has progressed or because they've had toxicity. By 4 months, only about 20% remain on trial. So the number or percentage of participants who actually have any kind of sustained response and not significant toxicity is actually very small. And so we have to be realistic about these numbers. This, by the way, note the caveat, adult participants, molecularly targeted agents. I don't think if we looked at pediatric trials, we'd see anything that was all that different from this, but I've never seen a curve like this for pediatrics. So interpreting the data on risks and benefits, the data that I just showed you, is not straightforward. First of all of these studies use surrogate endpoints. And second, and equally or perhaps more important, there are no controlled data here. There's no comparison groups of comparable patients who are not treated on phase 1 trials who may get whatever alternatives are available to them. And we'll talk about that a little bit later on. So I want you to imagine that we had a hypothetical cohort of patients who were eligible for phase 1 trials. And this is hypothetical data. My colleague, Julie Najida, a statistician, generated this for me just as an example, and so my gratitude to her. And so the median survival here, which is about what is published in sort of all-comer phase 1 trials, is 8 or 9 months. And just imagine that this is if you took patients and you just looked at their natural history and didn't enroll any of them on phase 1 trials. So imagine that you did the following trial, hypothetical trial, although I would love to see this done. If any of you really wants to be famous, if you do this trial and pull it off, you'll be famous. Imagine that participants give their consent to participate in a phase 1 trial. And so then you randomize them either to enroll in the trial or to be denied entry into the trial, so that you have a really fair comparison of entry into phase 1 trial versus not as an intervention. Not just a single phase 1 trial, but access to a whole phase 1 program, shall we say. And then you assess the survival and quality of life outcomes among these participants. What might you see, particularly with respect to survival? The bottom line is we have no idea. Nobody's ever done either a real experiment like this or a quasi-experiment or an observational study that tries to replicate this. So we really have no idea what would turn out. So you might see this, the blue being the control group, those who don't go on the phase 1 trials, and the yellow being those who do. You might see an adverse effect on survival. I've intentionally drawn this as quite small because I don't think it would be huge one way or the other. It's plausible. You might see no effect, entirely plausible. Or you might see some average benefit across the population. And I don't have any sort of ability to guess which of these would be the real outcome of this hypothetical trial. But in some sense, this plus the analogous quality of life question is the data that we really want to know to be able to say, do these trials offer a prospect of benefit to kids or adults who take part in them? The final thing I want to say is, when we're talking about interpreting data on risks and benefits, there are a number of challenges, some of which I've alluded to. One of which is, what do we compare this to? Right? What's the alternative case? What's the counterfactual? So do we compare these to access to licensed agents for these kids' condition or these patients' condition? And in this case, there are no licensed agents that are available because eligibility essentially requires that they have exhausted access to all of the licensed or really evidence-based agents for their disease, their state of relapse, et cetera. What about palliative or symptom care only? Well, clearly this is a reasonable alternative for virtually every kid that enrolls in a phase 1 trial. So clearly that is a relevant alternative, but it's not one that every kid and every family and every patient is going to accept. So the third possibility is receiving off-label, unproven therapies outside a trial. And I don't know how things go at Seattle Children's in the oncology program, but in both of the places that I have practiced, this is actually a fairly common thing to do for kids who have gone beyond the evidence-based first or second line therapies. So we have to be realistic and say, if the kid did not enroll in a phase 1 trial, what else might they get? And how do the risks and benefits of the phase 1 trial compare to the alternatives that they might get? I will come back to this point. All right, we've talked about risks and benefits. What about informed consent? So there are really good data that people who enter phase 1 trials, and I'll show you some adult data and then I'll show you some pediatric data, they enter in search of benefit, despite the fact that we've already seen that the prospect of benefit, while there, I've argued, is quite small, and that they misunderstand the purpose of these trials. So one study to point you to is by Chris Doherty and colleagues at the University of Chicago. This is an adult study where he interviewed 144 participants in phase 1 trials and asked them, why did you join the study? What's the purpose of the study as far as you understand it? And what do you expect for yourself in terms of medical benefit from joining the study? So here are the data. First, why did you join the study? Well, 73% said, I'm looking for anti-tumor response. And another 17% said, I'm looking for an improvement in the quality of my life. Of those who said, I'm looking for an anti-tumor response, 40% specifically said, I'm looking for cure or remission. This is a pretty ambitious or optimistic goal to have in mind if you're enrolling in a phase 1 trial. What do you think, what do you believe to be the purpose of this trial? 60% said the purpose of the trial is to look at how patients respond to the drug or how their cancers respond to the drug. And then a small additional number said they're both looking at toxicity and response. Whereas we've just talked about the fact that the main purpose of the trials is right there, determine dose and toxicity so that we can figure out how to take forward into phase 2. And do you think you'll get medical benefit from participating in this trial? 90% yes. So conveying a sense of optimism or hope about their participation in this trial. In some other data that I think complement this, Kevin Weinfurt and colleagues surveyed over 300 adults who'd been offered participation in a phase 1 trial at one of 5 major US cancer centers. And I'll show you, this included both those who had said yes and those who had said no, and I'll show you some differences between those two groups. So about 80% chose to enroll in the trial of those who had been offered. And he asked 2 questions. Number 1, what's the probability that the treatment will control your cancer? And number 2, what's the maximum benefit that you think you might experience from participating in the study? Here's the answer to the first question, what's the probability that the treatment will control your cancer? About a quarter said 50%. I just showed you that the most optimistic, or at least for a classic single agent trial, the most optimistic answer would be about 10% for any individual. And so you can see that very few people are down where truth lies. A bunch of them are up at 50%, which is probably like saying maybe, maybe not. And then there's actually a bunch of people who are even more optimistic than that, thinking that the probability that treatment will control my cancer is quite high. Trial acceptors were, not surprisingly, more optimistic than those who declined the trial. So the median chance of benefit that those who accepted the trial said was 70%, whereas those who declined said 40%. Even the decliners are optimistic, right? What's the chance that you'll experience a serious adverse event? Acceptors, again, were a little more optimistic, saying it was just 30%, and the decliners thought it was a bit higher, 50%. And what's the maximum benefit that you think you might receive from the trial? Here, 40% said the maximum benefit was cure. This is really rather optimistic. These are almost anecdotal or case reportable phenomena where somebody is truly cured by participation in a phase 1 trial. Again, even the decliners were quite optimistic relative to what the evidence would show. What about pediatrics? So parents also show substantial misunderstanding of trials. And here I'm going to rely a lot on Rick Kodish's very recent work involving interviews and audio recording of consent interactions with parents of kids participating in phase 1 trials. And actually, many of these including the kids themselves, which is some interesting additional data that I won't be showing you. So what they did was they audio recorded and qualitatively coded 85 consent conversations. By the way, Seattle Children's was one of the 6 sites for this study, and some of you may be aware of it or even have had some involvement with it. And then they interviewed 60 of the 85 sets of parents, a mean of about 5.5 days after the consent conversation, and asked them a bunch of questions about what they took away from their phase 1 consent interactions. So here's what they took away. They asked them what they understood about the purposes of the trial, and specifically with respect to drug safety, dose finding, and dose escalation. And you can see that about half of parents showed no understanding here in the blue part of the bars of the issues of drug safety, the fact that the trial was focused on drug safety. A little under half of the parents showed no understanding that the trial was focused on dose finding. And the vast majority of parents actually were not able to articulate an understanding of the fact that we use dose escalation in cohorts to try to get to a maximum tolerated dose and to try to identify toxicity. Conversely, minorities of parents actually understood the drug safety and dose escalation points. A bare majority understood the dose finding point. And when you added these all together, in their composite summary, they argued that only about 35% of parents actually had a complete understanding, about another third had a partial understanding, and about a third had no understanding of any of these aspects of phase 1 trials. Investigators' disclosures actually also can be incomplete or misleading. Here are the data from, again, the Kodish et al. Study. And they asked a bunch of questions about, based upon their review of the audio tapes. So they actually had data on what was said, what was discussed during the conversations. They coded these elements as either being present or absent in the conversations. And so they first wondered, well, what about the scientific and design features of the trial? How well are we doing at communicating these to parents and to kids? So the goal of the protocol, essentially as a research endeavor, was articulated in the vast majority of conversations. The fact that the studies used dose escalation was articulated in about half, as was the fact that the goal of the protocol is to find a maximum tolerated dose or a dose to take forward to future studies. The phenomenon of dose cohorts was articulated in about 40%. Dose-limiting toxicity as a endpoint of the study was articulated in only about a quarter. And the fact that we will not be escalating doses within participants, no individual will have their individual dose increased, was articulated in fewer than 10%. What about discussion of alternatives, which I think is a particularly important part of the decision-making around phase 1 trials, or really any trial? And only about a third of these discussions included an explicit discussion of the fact that no treatment or palliative or hospice care is an appropriate option to consider. And the point that there are no curative options for the cancers of children who are eligible for phase 1 trials was only discussed in about 1 in 6 of these conversations. So I think these authors said there's room to improve many aspects of our phase 1 disclosures, discussions, consent interactions. And this theme comes through in adult work as well, although I'm not actually showing you the data. There's a nice study from folks at the NIH that's now about 10 years old, where they reviewed consent forms, so what is put in writing and what is approved by IRBs in disclosing and educating patients and families about phase 1 trials. So they reviewed 272 consent forms for phase 1 trials that were opened during 1999. So now we're going back a few years. How many of these included a statement that the study involves or is research? Well, essentially all of them. Most of them actually did identify the study as a dose escalation, safety, or toxicity study. Fewer than half, however, clearly identified the procedures in the study that were research procedures and distinguished them from the clinical procedures. And almost all of them made unqualified reference to investigational agents as, quote, treatment or therapy, as opposed to the experimental drug, investigational therapy, or some way of qualifying it to make clear that it's distinct from evidence-based therapies that we might use in other areas of practice. And this is, I think, one of the themes that keeps coming up, is the sort of therapeutic cast or therapeutic frame that is used to discuss even phase 1 trials. And I don't think we've quite figured out the right language to use to describe them, or put in place the right language to use to describe these, to clearly convey to patients, parents, families, the experimental nature of what it is that we're doing. All right, so we've talked about risk and benefit. We've talked about informed consent. Let's think a little bit about fair subject selection. And is the selection, particularly of children with advanced cancers, a fair selection of subjects? Is this the right population to do these in? Because as you'll see, this has been criticized. So how to think, just backing off for a moment, how to think about fair subject selection. So I think it's violated, among other things, if selection unfairly burdens a particular subpopulation. And I think it's not just that it burdens a particular subpopulation, but there has to be some argument that that burdening is unfair. And this is particularly of concern if the population is somehow vulnerable. So are dying patients vulnerable? And this is something we have to think about. And are children vulnerable? I think we all would agree that, at least in the sense that children can't actually give their own consent, at least younger children, and are dependent upon their parents to give a kind of proxy permission for them to enroll in a study, children are vulnerable. One of the hallmarks of vulnerability is the inability to give your own informed and voluntary consent, either because you're in a coerced environment, let's say you're a prisoner, or you lack capacity, let's say you're a young child or an adult with dementia or something like that. So George Annas, who has written very critically about phase 1 trials, George is a lawyer and bioethicist at Boston University, has a series of articles in the 90s where he's really critical of them, and he invokes things like Nuremberg to kind of set the frame for what we're doing in experimentation with dying patients in general, and he really has phase 1 trials in mind. So he writes, subjects who believe that they have nothing to lose and are desperate because of their terminal illness should be disqualified as potential research subjects because they're unable to provide voluntary, competent, informed, or understanding consent to the experimental intervention with such a view. Now, he's got adults in mind, but I suspect if you said to him, how would you apply this to children or to parents of kids with these sorts of who are in these sorts of situations, he would feel even more strongly about it. I'm quite convinced that he would. And so I think we have to wrestle with this view. Is it so impossible to get voluntary informed consent from either an adult who is dying or the parents of a child who is dying that we have to be ultra protective of enrolling them in research? And I'd love to come back to that in the discussion in the question period. His specific policy recommendation is that research in which terminally ill patients participate as research subjects shall only be approved if, and here I'm quoting from him, the research, if it carries any risk, has the intent and reasonable probability, these are my italics, based on scientific data of improving the health or well-being of the subject or of significantly increasing the subject's length of life without significantly decreasing its quality. And he goes on to argue that phase 1 trials, at least classically, don't meet this definition, and so dying patients ought to be protected from being offered participation in a phase 1 trial. So now we have to ask the question, is selection of terminally ill children for phase 1 trials unfair? And according to Merriam-Webster, the definition of unfair would be, is it marked by injustice, partiality, or deception? So then we have to consider the alternatives, and leave each of you to sort of think about whether you think it meets this criteria of unfairness. So one alternative, of course, would be just we won't do these studies, right? I don't know how we would proceed in therapeutic development if we didn't do the studies. I guess we would just take our best guess at the phase 2 dose and sort of jump in and hope that we got it right. So don't do dose finding or safety studies of these new pediatric agents. Another would be to do them in different populations. One population might be adults. Well, in general, we actually do these phase 1 trials in adults first, unless it's a drug that's for a uniquely pediatric disease where there would be no adult analog. And in that case, you might actually go into children first. But in general, as I showed you with the Chrysotinib trial, the adult studies are done, the adult maximum tolerated dose is clarified, and then the pediatric trials are initiated starting at about 80% of that adult dose. What about doing these in children with cancer as at least in Annis's view, less vulnerable population? Well, that strikes me as a non-starter. And what about children who do have cancer but are not terminally ill? Well, I'll talk about in a moment the fact that there actually might be scientific value to doing that, but I think that that would be a problematic approach as well. It'd be very hard for a child who has evidence-based options to say, well, would you be willing to forego these evidence-based options and offer them instead a novel drug that has barely been tested in humans or in children? So I think there are some valid reasons, besides just the vulnerability or not including the vulnerability, that do justify enrolling terminally ill children in these studies. But we have to think very carefully about these. So we might argue, are there scientific considerations? Are there scientific reasons why you actually learn more from studying these drugs in a population of children with advanced cancer than children who don't have advanced cancer? And here I think the answer is no. I think if we weren't worried about the interests and the rights and the moral standing of these children, we actually might want to do these studies in children with earlier disease. One of the concerns in children who've been exposed to lots and lots of different chemotherapy regimens is that their tumors may be so hypermutated, have developed so many resistance mechanisms to the therapies that they've already seen, that they may actually not respond in the ways that they would respond if these drugs were used earlier in therapy. The other thing is that these kids have often had organ damage from all of their prior treatments, and so they may actually not be a great sort of example of the toxicity experience because of their prior exposure to therapies. So I actually think scientific considerations does not justify using these kids with advanced disease. What about consent considerations? Are there reasons why either the kids or their parents are better positioned to give valid, informed, voluntary consent? And here again, I think the answer is no. I actually take seriously Annis's concerns about vulnerability and about the desperation of parents, families, children in these situations. And so I actually think it's hard to make an argument that these children or the parents of children with advanced cancer are better positioned to give consent than those who are earlier in their disease trajectory. So where I think we end up is with respect to risk-benefit considerations. That I think if you compare the risk-benefit profile of entry into a phase 1 trial for a kid with advanced cancer to the alternatives that are available to that child, it's a more favorable sort of relative benefit than it would be for a kid who is earlier in their disease trajectory and has more options. But this is a complicated notion that I'm still working out, and I'd love some sort of feedback or comments on as a little bit later. So I think this is where the action is. It's really that the reason we choose these kids is that for them, the risk-benefit profile is as positive as it can possibly be compared to all of the alternative populations. All right, so just to summarize, what do we do with all this, right? I've tried to reassure you at least a little bit that these trials are ethically valid, that they have a favorable risk-benefit ratio, although it's complicated, but yet there are real issues related to risk and benefit and consent and the vulnerability of children and families in these states. So I think the first thing we have to do is be clear about the decision tree for that families go through in deciding whether or not they enroll in a phase 1 trial. We have to do what we can to enhance the risk-benefit balance, but realizing that as we push benefit, we potentially increase risk. And so we're trying to sort of balance these two things as best we can. And then always improve the process and outcomes of informed consent and also child assent, the engagement of children in decision making. So when you read about the process of decision-making for phase 1 trials, particularly from authors who are critical of them, the sort of conceptual model of the decision tree that they lay out is often like this, that the distinction or the alternatives that families are choosing between is palliative care alone versus enrollment in an early phase trial. And for many people who are critical, they argue this is a better alternative for these kids than going on a phase 1 trial. I think that's not what the actual decision tree looks like for most families. I think there's really two decisions that are made sequentially. The first, are we a family who wants no anti-cancer intervention given where we are in the kid's trajectory, or are we still looking for additional anti-cancer interventions? And if we're a family that's here, then what are our choices? And I think our first choice is off-label therapies, something that our oncologist just concocts for us, or maybe something that's been published in one or a small number of studies, or a case report, or something that's not really hypothesis-based research, or enrollment in an early phase trial. And I think this is the thought process that families generally go through in making these decisions. What about the benefit-risk balance? So the first thing, I've showed you that we already do this in most cases, is to start with the highest dose that's predicted to be safe. Now, if it's a targeted agent, again, as I mentioned at the beginning, we're not necessarily always looking for the highest dose, but there may be some in vitro studies or maybe some adult studies that give us some idea of what's likely to be the pharmacologically active dose, what's likely to be the dose that saturates receptors, that turns off activated receptors, that sort of thing. So we may have some ability to estimate the right starting dose. And then I think if we're using dose escalation, we do want to dose escalate as rapidly as is safely possible. Now, I won't get into it, but there are some Bayesian approaches that are complicated and statistically intensive, but that allow you, for each child who's about to enroll, based upon every experience of every child in the study as well as any prior experience, to make your best statistically driven guess about the appropriate dose for the next child, and to constantly update your dose levels based upon every piece of accumulated information. These are not used all that much because they are so statistically intensive, but I think they're very attractive, at least philosophically, as a way to optimize the dose for each individual child. Other cruder efforts at rapid dose escalation have been tried, and at least one meta-analysis shows that they're actually probably no better in terms of response rates and benefit. They are probably more toxic, and they don't actually save you numbers of participants, give you any smaller trials. So these cruder approaches probably don't work, but maybe the Bayesian approaches, which haven't been well studied in meta-analyses, do. And then finally, ensuring informed consent. And here I think we have to be really clear and complete in our descriptions about purpose, about dose escalation. We have to be very specific about the low probability of clinical benefit, and we have to avoid some of the misleadingly therapeutic language that I showed you in some of those examples earlier. And we have to clearly identify symptom-directed or palliative or hospice care as valid alternatives for these kids in these studies, and encourage them to think about that as an alternative. In fact, some people have talked about the idea that you might actually require that there be a palliative care consult for any child or any family who's considering entry into a phase 1 trial, to really make sure that that alternative is explored and seriously considered. I'm going to end just by showing you a template that Shlomo Koifman, who's now at Cleveland Clinic, and colleagues at the NIH developed and published for phase 1 consents, that I think has a lot of value to it. When I say phase 1 consents, I mean consent forms. And you can't read this, but it's a nice short 3-page consent form, consent template. I don't know about the average length of cancer clinical trial consent forms here, but every place I've been, they've been 20 to 30 pages. So if we could get them to 3 pages, that would be good. Of course, this doesn't include 5 pages of HIPAA language. So the statements are nice and clear, and I think get the right message across. What's the purpose of the study? We're inviting you to be part of a phase 1 research study. We're doing this trial to find a dose of a research agent called blank that would be safe in humans. Future studies, phase 2 trials, can then test whether or not this agent is useful against cancer. How can I benefit from being in this trial? The main goal of the study is to test the safety of the research agent. It's not to treat your cancer. The chances that this agent will shrink your cancer or help you live longer are very low. However, what we learned from this study will benefit society and future patients with cancer. Pretty good. What are my other options if I decide not to enroll? It's your choice whether or not to be part of this research study. If you decide not to join the study, you may be able to receive different research agents in another research study, standard treatment for your cancer. It's always a question of what this would be for patients who are eligible for phase 1 trials. Almost by definition, they're beyond standard treatments. Palliative care, or care to relieve any pain or other symptoms you're experiencing, hospice care, or no medical treatments at all. This is nice and clear, and I think gets the right message across. All right, just to circle back to the Chrysotinib study, what did we learn from the study? Well, the main thing we learned is that the pediatric maximum tolerated dose is 280 milligrams per meter squared. That's something that is useful to clinical trialists who are developing Chrysotinib or trying to develop Chrysotinib as a potential therapeutic agent, but not all that interesting to almost anybody else, right? We learned a bit about the toxicity. Nausea and vomiting were common but mild. Visual disturbances were also common but also mild and transient. Liver abnormalities occasionally occurred and were severe but rare and also resolved. And neutropenia was a rare side effect, also severe but also resolved. So this is, again, mostly useful to the trialists who are developing the next study, who now know what to tell participants or families who might enroll in that phase 2 trial. We also learned a little bit about potential responses. And here, one of the things that this illustrates is the complexities of doing phase 1 research and talking about phase 1 research in an era of molecularly targeted studies that are really based upon good understanding of mechanism, good laboratory research. We're in an evolving period of phase 1 research where today's phase 1 research doesn't look quite like what yesterday's phase 1 research looked like. So we have to talk about this by different disease and ALK status. So ALCL, this rare lymphoma, all of them were ALK translocated, and of the, I think, 9 participants in there, 7 of them had complete responses. This is pretty darn good. If you were somebody, if my kid had ALCL that had recurred after standard therapy, at this point, I'd be very eager to get them Chrysotinib as a parent. What about inflammatory myofibroblastic tumor, also usually ALK mutated? And here there were 7, and the responses weren't quite as dramatic, but just under half had partial responses. About 60% had stable disease that was maintained on at least 2 measures 4 weeks apart. So that's some evidence of benefit for that subset. Neuroblastoma, I actually think this was a bit of a disappointment to the people who were doing the study. They were hoping that in these at least known to be ALK-positive neuroblastomas, that the response rates would be very high. But you can see that I think there was only 1 complete response, a couple of stable disease, and most participants had progressive disease. So not the kind of benefit that those who went into the study were hoping to see in this ALK-positive subset of neuroblastomas. There's also a larger group of patients who had neuroblastoma but were not their ALK status was not known. And here there was just 1 response, a few stable disease, and a lot of progressive disease. And then there was a handful of other, or not a handful, a number of other participants not selected based upon particular tumor type, not selected based upon ALK status, and a little bit of stable disease and a lot of progression. So I think knowing this now, the intuition that you all had at the beginning that maybe the prospect of benefit was different depending on whether you knew you had ALK-mutated tumor or not was right. And again, this brings us back to the idea that some of these molecular hypotheses, if the prior data is good, if the laboratory's data are good, actually really are borne out by the clinical experience. So just to sum up, these are challenging trials in children, but you all knew that before you came into the room. My view, despite legitimate concerns about desperation, subject selection is not unfair. The benefit-risk balance of these trials is not necessarily unfavorable. This, again, my view, when compared with the relevant alternatives. And this is, I think, the critical point. And if you read anything about the ethics of phase 1 trials, you have to be thinking about, so what is the alternative that this author has in mind when he or she is making risk-benefit claims? And finally, I don't think there's any doubt that we can do a much better job than we currently do in seeking consent from families, permission from parents to enroll their kids in these trials. I will stop there and look forward to your questions and comments. Yeah.
What a nice presentation. Thank you.
Thank you.
I'm going to broaden the context. I, too, live in your world. But we're faced with these same issues right now. We are trying a number of new agents in North Africa to treat Ebola. None of those drugs have been proven to be effective in the treatment of Ebola, yet the desperation there is fairly high. So when that person comes to the United States, the opportunity to provide maximal supportive care and one of these new agents brings these directly to bear and along the way. But for the audience, we do this every day. The realities are that until 2000, fewer than 10% of drugs in children, for use in children, had ever been tested in children. The rare exception was, in fact, in oncology. With requirements that all new agents have a evaluation in children, that number has come up to around 15 to 18%. So most of the time, every day that you're writing a prescription, you're doing a clinical trial. Now, most of the time, those risks are fairly low, but in fact, you're involved in that in a lot of ways. And those of us who work in these kinds of centers are constantly looking at, oh, let me see the use of methotrexate for autoimmune disease. Let me see the use of sirolimus for benign tumors that are miserable, lymphangiomas being the prototype. How do we bring that knowledge, this extreme organization that we have in oncology, to the broader use of how do we look at agents in children for all those common things we're treating every day?
Thanks for that very small question. You made a number of points, all of which are well taken. One of them is that we're sort of constantly experimenting. If we actually said what proportion of what we do is truly evidence-based, and you can point to the 2 or 3 randomized trials that clearly show that what we're doing is the right thing to do and safe and effective, all that, it wouldn't be the majority of what we do. So one answer, as you probably know, there's been this discussion about learning healthcare systems and the idea that we should be using our systems, and particularly now that we have much better IT systems and electronic health records that are starting to be things that we can use to collect data from, to learn from every patient experience. And so one answer to your question is to use just a routine practice of medicine where we are using things that are off-label or not well validated to begin to answer questions about, well, what happens when we do that? What happened the last 5 times we did that? So that's part of the answer, is there's a lot of this just going on and we can't do controlled trials for all of it, so let's do what we can with the observational data. The other is to say, where we can, let's actually do clinical trials of these things. And if we know that we're going to be using something broadly, let's do it in a way that we can learn from. And so I think there's 2 answers. One is to move as much as we can into the research arena, but the second is to realize that we can't do all of that. Let's learn from what we can from what we can't actually study prospectively. Ben.
Great. Thank you, Steve. That was terrific. I want to get some clarification from you at the end of your talk with your 3 issues of that you thought were satisfied for clinical trials. The very last slide. So my question, though, is how do you relate those 3 points about subject selection, risk-benefit, and consent with the issue in the very beginning of prospective direct benefit, and in particular, using the example of your trial where they were enrolling some kids who did not even have that marker? Would you consider them to have been offered a prospective direct benefit, and do you think that regulatory concept fits or squares with these 3 criteria here?
So I think it's a very difficult call. So one view is based upon what we know from the laboratory experience, based upon what we know from the adult experience, we know the drug, we know its mechanism of action, we know its target, and we should really only be taking it forward in patients who have that target, where we can claim based upon reasonable mechanistic data that there is a prospective benefit. Another view is there's a lot we don't know, and there have been a number of quote, targeted oncology drugs studied that end up having, quote, off-target effects. And if you really only limited them to what you thought were the targets, then you might miss these other pathways, these other potential areas of response. So I think it's a struggle in the oncology community right now is, do we limit our earliest phase studies to situations where we know the target and we can identify the target? Or do we remain open to the possibility that there are other as yet unidentified targets, and so explore the drugs more broadly? And then if we see a response, let's say in a kid who's not known to have one of the targets, then you dive deep into that tumor and you say, what is in that tumor, or what other mechanism of action might there be? Because I think as the idea of molecularly targeted agents or precision medicine sort of conveys the idea that we have a sniper rifle, but in fact, it's not quite a shotgun at this point. It's getting better than that, but it's not quite as accurate as a sniper rifle. So that's the struggle, but I think the point you raise is a really good one. Yeah.
Hi, Steve. Thank you for that talk. I'm Bruder Stapleton. I'm a nephrologist, not an oncologist. You didn't touch on assent, whether it's required, if it should be required, at what age, what developmental stage that should be, if it is something you think is important in this. That was one question. And second question, would it be ethical to take this agent to a phase 2 study for neuroblastoma, given those results?
So let me answer the second question first, because it's a shorter answer, and then we'll come to the first one. So I think it is ethical, and in fact, I think the COG may jump over phase 2 and go right to phase 3, interestingly, and they may also move it up into earlier phase treatment, not just recurrent disease, in combination with other agents. Now, just for kids who are known to have ALK-mutated tumors, so not all comer neuroblastomas. I don't think there's going to be any further study of the drug in kids who don't have ALK-mutated neuroblastoma. So, and the argument for doing that is that although it may not have had much response as a single agent in these kids with very advanced disease, it's hard to sort of generalize from that to what's going to happen when combined with other agents in multi-agent therapy early on in disease. So I think appropriately it's going to be moved into early therapies. Now there are sort of arguments about how do we do that, what's the best sort of model or study design, but I think people are not going to drop Chrysotinib as a potential agent for ALK-mutated neuroblastoma. Your question about assent, my own view is that in phase 1, so there is language in the regulations, as you probably know, that assent is required from children who are capable of giving assent, with a lot of sort of fuzziness around who's capable or not. There is this exception that says if there's a prospect of direct benefit to the child from the research that's only available in the context of research, then assent is not required. I understand that, but would like to interpret it quite narrowly. And particularly with respect to phase 1 trials, where the kids often have a lot of experience, they understand their own situations, they often are mature beyond their years, and they have sort of really fundamental choices before them, kids and families do. That for me, it's the rare phase 1 trial where I would want to take at least a middle school or older kid and say, you have to be in this trial again over your objections. Now, occasionally, if there might be something that was just a dramatically effective drug in adults and the same tumor occurred in children, and we were now studying in children for the first time, I could envision exceptions to that rule, but I think the general rule is kids who are capable of assent, and again, lots of questions around assent, should only enroll in phase 1 trials with their agreement and assent. That's my own view. Yeah.
Very nice, crisp talk. It helped me think about this. I'm one of the oncology folks here. One statement, or at least a point of view, I think, is that this contrast between palliative and hospice care and participation in a phase 1 trial or any other thing, I think that's a bit of an unfortunate dichotomy. I think that really we should be always primarily concerned with this phase of patient with the palliative and hospice, and that might include the participation in a phase 1 trial. In some ways, I think offering a participation trial that stands in contrast to that is completely the wrong approach, would be my opinion about that.
Yeah, I completely appreciate the point, and I think you're completely right, and I think I didn't do justice to that in the presentation, and actually I should probably modify this figure to show that if you don't go this route, which would be palliative care alone, that this should be anti-cancer
And.
intervention plus palliative care, off-label therapy plus palliative care, early phase trial plus palliative care, and that would be a better figure.
I completely agree. The other comment or question, I guess, is it seems to me that while the potential benefits in general that we can ascribe to a phase 1 participation in a phase 1 trial are relatively constant. You showed the data. We don't know ahead of time,
Yeah.
but the potential risks are quite variable, and there may well be that ratio might very well determine that a phase 1 trial is really not ethical for certain children. I think, fortunately, as we enter the molecular targeted therapy, most of the time we have less toxicity to deal with. I think that was much more problematic when our phase 1 agents were chemotherapy agents.
Right, but it's interesting, but then you have the examples of CAR T cells, which are both incredibly risky and also probably more potentially beneficial than many of the phase 1 type interventions
Right,
we've seen in
right.
a long time.
That's right. That's absolutely right.
So unfortunately, we are past 9 o'clock, so we have to end. But thank you, Steve, for your presentation. Thank you all for attending. That's great.
Thank you.
Very good.
Thank you very much. Yeah, hi Russ, how are you?
So, Abby and I were talking about this the other day. One of the things that does trouble me a bit, having participated in